evening again and welcome to Genetics and Archaeology, DNA Research Changing Human History. We are happy that you're able to join us today to learn more about how DNA research is changing our understanding of human history and the rise of civilization in the Mediterranean basin and in the Middle East in biblical times. My name is Natalia Gines, and as Associate Director with the Global Engagement and Affinity Programs Group at UChicago Alumni, I have the honor and the greatest luck to support UChicago alumni that work or reside in the New York City area. Before I introduce our moderator and speakers, I want to go over a few quick logistics items so that you are able to fully participate of the session today. As Andrew had been talking to a few of you, all of you will have been muted and will remain on mute during much of the presentation. We will have two opportunities throughout the session for you to ask questions from the panelists. You can submit your questions through the chat space accessible through the menu bar that appears at the bottom of your screen. Once you click on it, you'll be able to type your question on the spaces to the right of your screen. So make sure that you start the message with the word question. There's a lot of you, I believe at this point it's over 80 participants. So if we can see that initial word question, we'll know to grab it and place it on our list. And the name of the panelists that you would want to direct your question to. There's a lot of you, as I said, so we will try to select a few questions midway and at the end of the session. You can also, at the bottom menu bar, you can raise your hand if you have a question and we will try to get to you as well. And at that time, we will unmute you and you can ask your question as well. The session is being recorded, as I said, and a link will be sent to all of the registrants and participants within the next few days. Now, without further ado, I would like to introduce our moderator, Andrew Peters. He is a 2011 MBA graduate with a keen interest for archaeological research and one of the people responsible for making this session happen here tonight. Andrew?
Natalia. So until last year, I was president of the Booth Alumni Club of New York City, and I've been really passionate about archaeology just on the personal level. So last year, seems like a long time ago, last summer, I took my son Kevin on an archaeological dig with Professor David Schloen and Sandy Schloen, who are on this call today. So that's part of the backstory. So happy that we have so many people. I wish we had a happy hour right before or after this, but maybe we can do that in a sense as well. Yeah, I really have an exciting lineup of four eminent professors, eminent speakers tonight. So we have the chance to really talk about a collaboration across two very distinct disciplines between genetics and archaeology. And this is something that can really only take place at really a top-class research university. So from my point of view, this is really about looking into an entrepreneurial exercise that's taking place within academia that we as alumni are really well positioned to support. In fact, a lot of times the gaps in terms of funding for these types of projects are really hard to find. I think alumni can take up the slack in looking for some of these sort of difficult but very rewarding projects to help with. So our first speaker, John Novembre, he's a professor at the Department of Human Genetics. He's really been at the forefront of research in the human genome. He's been asking questions about hereditary disease, genetic basis of human evolution. And he's really important in terms of the collaboration between genetics and archaeology here. We also have Maanasa Raghavan, assistant professor in the same department, who has been working on questions related to how do populations and societies, how do they transition into the present day populations that we see? What are those different factors, demographics, environmental, that impact that? And so a lot of really cutting edge research that she's doing, she'll be able to talk about. And then from the Oriental Institute, the OI, we have David Schloen, the professor that I mentioned before. And he's been doing a lot of things, but one of the things he's been looking into is the Phoenicians, and it's a sort of well-known, very rich trading culture, but hasn't had as much done work on it as other ancient civilizations, and he's been doing work on that. And then we have Sandy Schloen, who I also met on the same dig, and she is really thinking about how do we capture all this data that's generated in the process of archaeology, including genetic data. How do we put that into a database and that it's available and accessible to future generations and future researchers? So this is a really important part of this project we'll be talking about tonight, is how do we take this very difficult to access and analyze data and how do we sort of put this puzzle all together. So that's very interesting to me from a couple points of view. So as an analytics guy, I really appreciate the difficulty of having clean data, and there's probably nothing dirtier than archaeology in the sense of you don't have anything to go on except for what you see and the context of where you find it. So I think in the genetic side is really almost like an archaeological dig in the sense that you really have so much, places to go, and you have to sort through how do I start and how do I put this all together into the context and how do I actually learn something from it. So without further ado, I'll pass it over to John to start at this point.
Great, thanks Andrew for the introduction, and thanks all of you for coming together to hear about the work that we're doing in this exciting field. I'm going to start us off with an introduction to ancient DNA research. Can everyone see my slides? Andrew, can you give me a thumbs up if you can see my slides?
Yes.
Great. Okay. Thank you. Super. So I wanted to start with a particular puzzle that is from North America, the Kennewick Man. This is a skeleton that eroded out of a riverbank on the near the town of Kennewick, Washington, in the Pacific Northwest. And the original coroner who looked at it thought that it would be an early European settler in the region. And but then radiocarbon dates were taken from the sample, and it turned out to be 8,000 years old. And the certain features of the skeletal morphology led some of the earlier anthropologists to emphasize that this looked like a European descendant individual. So what was a European descendant individual doing 8,000 years ago in the Pacific Northwest, right? This is a time when it was thought that Europeans weren't there. So this led to a lot of controversy, people asking, well, was it really European? Maybe it's actually Native American. And a large struggle ensuing actually over this sample, and actually who should have the rights to study it. Should it be reburied according to the wishes of the local Native American people? Should it be kept for study by local anthropologists? And the bones were originally discovered in 1996. This is a cover in Newsweek that covered part of this controversy. And so I want to come back to how this controversy was resolved through the tool of ancient DNA. But before I do so, I want to introduce what is ancient DNA. Okay, so this is research that begins in the field, in the archaeological sites, the kinds of work that that David and Sandy, Professor Schloen, do here at the University of Chicago, and where bones are taken from the field with very carefully measured and lots of auxiliary data collected, or from museum samples. So these are jaw samples where teeth are being extracted, because teeth are one of the tissues in the human body that can preserve DNA very well. Inside of the tooth, the roots, you can it holds DNA well, preserves it for thousands of years in good cases. In the lower right here is a petrous bone, another bone that is known to preserve DNA very well. These are pictures from work that I'm involved in at a research site in Sardinia. So after the samples are taken from the field, from the museum, they're brought into an ancient DNA lab. These are sketches from for the design of the ancient DNA lab that we sought to build at the University of Chicago during the planning phases. And actually now with our successful recruitment of Maanasa Raghavan, who you'll hear about next, she has actually finished this design and built out an ancient DNA lab. She'll say more about that in a minute. But these are lab spaces that are extremely clean because you want to keep any modern human DNA out of the sample. Our DNA is such high, in such high molecular concentration that it can easily confuse all of the chemical reactions that are used in trying to extract the small amounts and broken up fragments of DNA that are preserved in these bones that are being taken from individuals from thousands of years ago. So from the lab, it's prepared for DNA sequencing, and that involves isolating the DNA from the cells. So this is a schematic to remind you about your DNA. Each of us inherits 2 copies of each piece of DNA, these chromosomes, one from our mother, one from our father. And if we look at these, they are composed of a sequence of base pairs, and we can look at the same base pair in multiple individuals and ask whether they're the same or different, and use those similarities and differences to learn about how people are related to one another, and in turn, how they've been moving across the planet, contacts between different populations that allow us to learn about the past. And so this is a variant locus, we call it, with we'll call it a C allele and a T allele, because we usually just read one of the 2 strands of DNA. Okay, so here we're reading the red strand, the C allele, the T allele. Okay, and this individual we would say is a CT heterozygote. Okay. So how do we actually read this DNA though? The way that it's done is that the DNA sequence, so if we imagine the letters that run along that red strand in the previous slide, they're converted with chemistry into fluorescent dyes that will trigger a sequence of light flashes in a sequencing device. And there are different fluorophores used for the different bases. So an A might show as a red, a C as a blue, a G as a green. And the sequence of light flashes can be read using very high-powered cameras, the same kinds of cameras that are used in high-grade astronomy and for detecting distant stars. We're actually focusing these downwards on slides and looking at the sequence of the flashes to read out the sequences of millions of DNA molecules at a time. And these images are then reconstructed to make an inference of what the sequence is. And what's been incredible is over the last decade or so is that these machines have become incredibly efficient. So they can read, for instance, 250 billion bases in a 25-hour run of the machine. This makes ancient DNA research possible because you can sequence an old bone that is largely going to be full of DNA from the bacteria that have infiltrated the bone from the soil. But amongst all of those reads, there'll be a few human reads that we can sift out and use to analyze and study human history. When we're done, we end up with data files that look like this, where each row is an individual here, and we have some individual level information. This is very simple, just 5 little, kind of tags representing like individual IDs. Sandy will tell you about a lot of the complex sort of interfacing with databases to have rich information about the architectural context of individuals. But what I want to focus on here is the genetic data, which is each pair of letters here represents the genotype of an individual at a specific location in the genome. So this is about 10 locations because of these different columns, of these pairs of columns. And then, as I said, each row is an individual. The real scale of these data sets today is such that we will be looking at millions of columns expanding to the right here, and often thousands of individuals. So there are huge bioinformatic challenges, and that's actually my expertise. I'm a computational biologist. And so I analyze these types of matrices to try to learn about population relationships. One of the tools we often use is something called principal components analysis. And it's a way of taking these matrices where rows are individuals and columns are what we call single nucleotide polymorphisms. And here we encode them numerically rather than with the letters that you see here. And we can use this tool of multivariate statistics, principal components analysis, to compress those millions of columns that I described down to just say 2. So we look at 2 numbers that summarize all of the information in all of these columns. And then we can build plots that show for each individual where they fall in this 2-dimensional space. So this is a kind of genetic space, the positioning of individuals in genetic space, and the relative relationships of individuals in that genetic space can tell us about the past. So here we see 3 clumps that might indicate to us that there are 2 source populations with a set of individuals who are mixed in between them. In this case, I simulated the data. I know that's actually what should be happening, and indeed that's what we see. Okay, so how has this tool helped with resolving the Kennewick story? So this is a story I read about when I was just finishing high school. 1996 was when I was finishing high school. And I was fascinated with the human past even then. And in 2015, I read about a group doing ancient DNA research where they had extracted DNA from these bones and looked at the DNA to help understand the origins of this individual more clearly. And it was a big surprise to me. In 2016, I got a call from the U.S. Army Corps of Engineers, a kind of surprising call, not the normal kind of call you get. And they were asking me, would I be willing to reanalyze the data generated by this team? It's actually a team in Copenhagen where Maanasa, Professor Raghavan, who's speaking next, trained, one of the top teams in the world. And if I were willing to reanalyze the data to help confirm their results. Okay, so this story that I read about as a teenager, as a professor, I'm now actually being asked to weigh in and give an opinion on the answer to this. It was really exciting for me. And this is the figure that we generated in doing that reanalysis of the original data. And it's one of these PCA maps. And here you see Europeans so these little abbreviations are individuals. And the big circles represent like a summary of sets of individuals. But you can look at the big text here. So Europeans are descendant individuals are largely in this lower part of the graph. Native Americans sampled from today are in the upper left-hand part of this graph. East Asian, Siberians are kind of along this axis here. But importantly, the Kennewick man, this sample that has been so controversial, just clearly comes out as Native American and has no evidence of being European at all. So that settles this debate once and for all. And it was very powerful for me to hear about a debate that had been running for 16 years and to finally have some resolution thanks to science, right? And thanks to advances in technology and these new techniques. So soon after this, actually, the Corps of Engineers made a final designation, and then the sample is actually returned to Native American groups. Okay, so I want to tell another story about the power of ancient DNA, which is based on work that I did in Europe, where we looked at a large number of genotypes in about 1,400 individuals from Europe. And they were mainly sampled in London and Lausanne, but we had information about where their grandparents were from. And so we built one of these PCA maps. And here I'm showing that each two-letter code is an individual and an initial view of the results. Then the large dots now represent the median position of all individuals with the same two-letter code. Some of you may recognize these as the ISO country codes and already be recognizing the patterns here. Well, let's compare it to a geographic map that has a legend for those codes. What you see is in this map is that the genetics of Europe are a lot like the geography of Europe. That here you can see Spain and Portugal and France, Italy, Switzerland, Belgium, so on and so forth. So there's a large, a strong relationship between the genetics and the geography. A lot of little wrinkles I can tell about this map, but I just want to emphasize this broad pattern. And one of the questions that I would get about this after we published it, and you should say this was very exciting when we found it, because it showed how with a large number of measurements across the genome, you could see fine scale structure even in a very from a genetic perspective, a very homogeneous population. So the question we got was, well, if this is the pattern today, how long has that pattern persisted? Is this what the genetics of Europe had looked like 10,000 years ago? Or is it something that's just something that arose over the last 2,000 years? And so when did these patterns become established? And this was one of the first hints that actually these patterns may not be that old. This is an image of the Tyrolean Iceman, a man who thawed out of a glacier in the northern Italian Alps near the Austrian border, and his DNA was one of the first. It was one of the first humans, ancient humans, to have their DNA sequenced, the whole genome, and this is where the Iceman falls within that same genetic map that I generated. So he lands over here by these are actually Italian samples. Let me go back a slide. And you'll notice there are these 5 Italian samples over here. I need to move things there. These 5 Italian samples, kind of curious to us in the initial analysis, because they don't cluster with the other Italians. It turns out they're Sardinian, we found out later, okay? And when we look at the Iceman, he clusters with these 5 Sardinian samples. So does that mean that the Iceman was a Sardinian on vacation in the Alps in the Copper Age, 5,000 years ago? That's the radiocarbon date on this individual. The suspicion was not. That actually what it reflects is that there's been a substantial amount of change in mainland Europe within the last 5,000 years, and that the ancestry of this Iceman is somehow preserved in Sardinia. Okay, and that's part of why I've done more research in Sardinia. And this hint that things have changed on the mainland has been developed extensively since this first study. And the view now, thanks to ancient DNA that has emerged, is that Europe is the mixture of early hunter-gatherers, represented on the left of this nature cover, then farmers coming in during the Neolithic period, represented by ancestries like the Iceman and the sample from Stuttgart, Germany. And then later, a mixture of what were first called ancient North Eurasians, represented by this MA1 sample, a really important ancient DNA sample that actually Maanasa, Dr. Raghavan, who's going to speak next, was the lead author on the study that published the MA1, because what that revealed was that there's been an influx of ancestry to Europe in the Bronze Age period relatively recently. And so those patterns that emerge of seeing this genes mirror geography in Europe, that PCA plot that I just showed, are actually the result of these 3 groups mixing over time until geography is the main determinant. Okay. So that expansion occurred in the Bronze Age. Just sort of some figures illustrating it. I won't detail this, but there's a pair of influential studies that I helped review in 2015. And overall, like an emerging view that is coming out of these ancient DNA studies is that many of the clinal patterns, these like relationships between genetics and geography that are observed today, actually seem to have arisen from the superposition of many like discrete punctate population movements rather than stable continual gene flow, which is more the model that dominated when I was in training. And so this ancient DNA research has really forced us to shift how we think about the human past. It emphasizes a really highly dynamic nature to human history. And that means that there's a lot of opportunities to discover and detail that dynamic history. I like this quote from Joe Pickrell and David Reich. Ancient DNA results are so regularly surprising that almost any measurement is interesting. New historical discoveries have been made in virtually every ancient DNA study that has been carried out. It's a strong statement, but it bears a lot of truth. It's just been really amazing to see what we've found with ancient DNA research. From the Kennewick Man to the Tyrolean Iceman as just sort of 2 examples that I've shared here. But so I hope that's a decent introduction. Now, Dr. Raghavan is going to tell you more about her research. She is a top ancient DNA researcher that we've recruited recently to the University of Chicago. She's helped build our ancient DNA lab, and I'm excited for her to have the chance to share her work with you. So I will stop the screen share and let Maanasa take over.
Okay, is that visible? Can you see my cursor move? Yes?
Yep, yes.
Perfect. Okay, thank you. Thank you so much, Professor Novembre. It's a pleasure to talk to all of you. I'm very excited to spend the next 15 minutes or so telling you a little bit about some of the practicalities and really bringing this kind of research closer to home. was just Chicago for some of us. So I'm going to take a few themes off of Professor Novembre's lecture. Particularly want to tell you a little bit about what it takes to do this kind of research. Why do we need these kind of facilities, and what do we have here at UChicago now? And really what that enables us to conduct in terms of research themes, as well as hopefully if I have time, I can get into a couple of examples of projects that we're in the process of setting up. And I say in the process of because I'm a new recruit, so I just started a couple of years ago. And the first year really was spent sort of putting on a hat that I never thought I would, which was part architect. So as Professor Novembre said, a lot of this research requires this really sterile facility. And so to kind of build that from scratch was really exciting and sort of, I guess, pulled a lot of creativity out of me in ways that I wasn't aware of prior to coming here and starting a new lab. So I just wanted to give you a sense of why we need why we need an ancient DNA lab and what is it, how is it different from any other standard molecular biology lab. And to kind of make that point, I want to take you back essentially to the advent of ancient DNA. I found this really interesting figure online, and it's sort of derived from something called a hype cycle, which apparently is something that is used in IT, information tech, to essentially set up how a particular new technology matures over time and what are the different stages as it does. It's pretty controversial, but I found that somebody had overlaid the progress of ancient DNA as a technology over time, and they sort of got it bang on. So essentially what we're looking at here is the initial sort of innovation that sort of set off this ancient DNA field. And this was back in 1984, where the first ancient DNA paper was published. And so since that started, it sort of led off to this explosion, that little spark. And for the next about 10 years or so, what they call the peak of inflated expectations, all the way from people publishing in top journals that they had sequenced DNA from Egyptian mummies to even dinosaurs. And then very suddenly towards the mid to late 90s, all the excitement sort of waned. And that's because we started to understand better the molecular characteristics of ancient DNA, and the fact that a lot of these big studies that came out in late 80s, early 90s couldn't be replicated, couldn't be trusted, and I'm going to get into why that was so. But fortunately for us, people didn't give up, and so while we're at the bottom of this, trying to figure out what went wrong, that sort of sparked off this new sort of avenue and this idea that we can still do it, we need to take care. And so that set us off on the slope of enlightenment to recognize what the limitations of a field can be, but learn from that. And so we're sort of now, I would say, in this phase, or we're still in the slope, but apparently at some point we're meant to reach a plateau, but we're still far from that. And as long as we have samples, as long as we have creative people thinking and doing, and of course funding that supports this kind of research, I think we have a lot to still answer, a lot of unanswered questions. Right, so as I mentioned, just trying to kind of focus on what happened and what we learned in this period that set off this new spark that brought us to where we are today, is the fact that we got to know what the molecular characteristics of ancient DNA are. And there was a seminal paper that got published in 2000, which is sort of a stern warning and sort of warned people to do ancient DNA rightly or not at all. And they set out these criteria for how you could be a good ancient DNA researcher. And I've highlighted two that are very specific to ancient DNA. A lot of the other points are sort of general, if you're a good scientist, a good molecular biologist, you should probably do these anyways. But the first and the third point are very specific to people doing ancient DNA research. And so one of those novel understandings of what ancient DNA was the idea that there was certain molecular behavior with ancient DNA, and we need to recognize that and understand the limitations of that, so we can then do the work in a solid kind of fashion. And sort of stemming from that was the fact that once we recognized those limitations of ancient DNA as a template, what can we do to make sure that that doesn't impact the quality of the work? And so one of the first sort of criteria that this paper laid out, which everyone today follows, is this idea that we need an isolated, sort of almost forensics grade facility to do this kind of work. And so just dropping into what it means by molecular behavior of ancient DNA. So, these, as John's presentation laid out, a lot of these samples, of course, come from archaeological records and archaeological contexts. So these samples have been lying in dirt for ages, for tens of thousands of years. And consequently, a lot of environmental factors get to the DNA backbone. And these arrows are just meant to show you the different locations along your DNA molecule that are susceptible to damage. And there's a lot of these, which means there's a lot of ways the DNA can get damaged over time. And so one of the big ways the DNA can get damaged is over time, your intact DNA, if you're living, can get chopped up once you die. And that's, again, thanks to all these different factors. And so as DNA degrades, it gets chopped up into tiny, tiny bits. And over time, even those tiny bits disappear from detection limits. And so it's fragmented ancient DNA compared to modern DNA, as well as limited in quantity. The other issue with ancient DNA compared to modern DNA is modern DNA is pretty pristine. When you sequence it, you get what you read. Well, with ancient DNA, because again, it's been lying in dirt, sort of susceptible to all these different environmental features, it starts accumulating mutations that are not biological. So as an example, on top here are the different sort of DNA bases, so ACTG. So this is just a random example of a code that you may be reading and what you may expect from a human. But as an example, these are sort of examples of the DNA that maybe somebody's extracted from an ancient sample. And what happens here is you may get a position where all your reads are sort of, they develop a mutation. And usually if this is the case compared to what you expect, you would call it a biological difference, a true mutation. But sometimes the ancient DNA, you can also get spurious changes, artifactual changes. So for instance, here, in this case, most of the other fragments that you've sequenced from that individual are what you expect. But then there's one that changes here. Similarly, there's one here in this position where you expect C's, where you have a single T. And so once you start seeing these sort of random changes that are not consistent, that would imply that these are likely not to be biological mutations. And so you have to be very careful and take this into account so it doesn't filter down to your downstream sort of analyses and inferences and cause skewed inferences. And so one of the big consequences of the fact that ancient DNA can be damaged is that you, particularly if you're studying human ancient DNA, that you as a human are your sample's worst enemy. And so as an example for this, I'm bringing back this lovely plot here. And so some of these early studies of Egyptian mummies, for instance, were carried out by two renowned ancient DNA researchers. I mean, both Alan Wilson and Svante Pääbo have done seminal work in this field. But also some of the early work came out of their lab. And today we know that some of the Egyptian mummies that were sequenced and published in the 80s were actually Svante Pääbo himself. So it was contamination from the researcher. And so when your samples are so precious and susceptible to contamination by yourself, and not just by yourself, but also potentially archaeologists and upstream and students who may be working with the samples in the lab, you need to be very careful. And this is when point number one, which is isolation of workspace, comes into play. And so ancient DNA facilities are isolated. They're distinct from normal molecular biology labs, and which is why it's an added investment on the part of any university that wants to develop and sustain this kind of research. So I just want to quickly give you an appreciation of what we've developed in UChicago over the last 2 years, although the story actually extends well past my arrival here because Professor Novembre and many other colleagues in human genetics actually started the groundwork for this quite a few years before I arrived. But just before we get to Chicago, I just wanted to give you a sense of where we're at today in terms of ancient DNA labs globally. And so you can appreciate that there are clusters, particularly in Europe and North America, again, reflecting funding status and universities and institutes that are able to sustain this kind of expensive research. But also, I have to say, unfortunately, a lot of these labs, in fact, tend not to publish too much. As Professor Novembre mentioned, there are 3 or 4 labs in the world, 3 that I have highlighted here, which really generate about more than 95% of the studies and the knowledge that we have today, at least the last 3 or 4 years, within the field of ancient DNA. And there are many reasons for why there's almost sort of monopoly, if you can say, in terms of which labs are producing in different continents. And it does come down a little bit, as I mentioned, to funding, but also there's an important point here, having again trained in one of these big labs, is the idea that what sustains this kind of research, which is really at the crossroads of multiple fields. We're looking at archaeology, anthropology, genetics, computational biology, is the fact that if you can build sustainable multidisciplinary collaborations and keep that going over time, it really helps kind of boost this kind of scientific creativity that leads us to ask these exciting questions about our past. And of course also gives us access to samples. But if you're not able to build that, then unfortunately this kind of research can't go on for a long time. So it's very important factors to keep this kind of research going in any location. So simply building a lab is not enough. And so coming back close to home, I just wanted to give you a sense, and hopefully this Google map is familiar to a lot of you if you've been on the campus at some point, that we do, one of the big issues when it comes to building a new lab is space. And luckily we were able to find a space in the Gordon Center for Integrative Science. So this is Ellis and 57th, for those who are aware of where that is on campus. This is the medical center. The Cummings is where our modern DNA lab is. And so like I said, it has to be physically isolated. So this is a good 3-minute walk down the road, which is not a lot. So it's still great for being in the office and then popping over to start an experiment, but at the same time, make sure that there's less contamination from modern DNA sources into our ancient DNA lab. And just to give you a sense of how it's sort of set up, it's about 1,400 square feet. And sorry if I sound materialistic when I put this number up, but the reason I wanted to put that up is to give you a sense of how expansive the lab is, that we could actually isolate it into different compartments, so self-sustainable compartments with their own ventilation, their own decontamination setup. So we have an office where students can sit when experiments are running, but then we have 4 different lab spaces that have that are sort of self-isolated. So 4 different students can work on 4 different projects at the same time without contaminating each other. And this is really important because it means that we have high throughput or high capacity to conduct multiple experiments at the same time. And just to give you a sense, so when you walk into the space, you can either go to the office or you can get into the lab space where you have to gown. So again, because of the sterile conditions, you have to be fully gowned all the way from top to bottom. And then you enter and you get into what's a communal space with sinks and freezers and fridges where we store our samples. And then you sort of start getting into individual lab spaces, which are fully equipped now. It took us about a year, but everything's in place now to do all kinds of projects. And just put out there, we're also celebrating, of course, we're not all about work, but we also celebrate our small accomplishments. So that was the day of our Ancient DNA Lab inauguration last year. So I don't know how I'm doing for time. Okay, so I guess I have a couple of minutes. So I just want to maybe fold these together to say that a lot of our work is just beginning because, as I mentioned, the lab's just about up and running now. So we've started a lot of exciting projects. A lot of our projects are very much along the lines of what Professor Novembre has talked about, understanding human population histories by linking the genomic diversity of the past to the present. We're also on the side interested in following other species. So we also work with archaeologists, including people on campus, to understand other species understand other species evolutionary history, so domestication. And let's see, I'm gonna probably skip this, but go on to just the last few slides where I wanna give you a sense of exactly what projects are ongoing. So just to give you a sense of how many ancient DNA human data sets are available all through the world as of 2018. Most of them, the red dots are ancient DNA samples that have been published. Most of them come from Europe. There's a lot of different continents even that don't have data. And so a lot of our work is involved in sort of answering questions about places that have not been studied yet. And so one of the big sort of projects we have ongoing in South Asia, where we're trying to link sort of cultural and genetic goings on in the other parts of Europe and Asia with South Asia, and try to understand the causes and sources of different ancestries. And we do this using both ancient DNA, this is me visiting an excavation, as well as working with local communities to kind of understand the past, and link the past to the present. And then the reason why I put this project is I wanted to highlight the fact that UChicago is also helping us support, research in global locations. And so a key backbone of this project is the center in New Delhi is a part of the global center that UChicago has, as well as the South Asian studies on campus, which links us to historians and archaeologists and anthropologists who study South Asia on campus. And that's really important because, as I mentioned, ancient DNA is interdisciplinary, and it links really three big fields together. And it's really great because, in fact, having a panel discussion like this with, starting with John, Professor Novembre's group, which is computational, my group, which is more empirical and does molecular biology, and then to Professor Schloen, who's with the archaeological experts, have all of this on the same campus means that we can start actually doing interesting stuff on campus. And so one of our newest studies is actually in collaboration with the Oriental Institute, where we're trying to kind of see some of the same ideas that Professor Novembre talked about in terms of migrations into Europe and how the European landscape is set up. And so one of the sites we're looking at right now is in central Turkey, where we're particularly interested in understanding what happens in this transition, the cultural transition that happens about 3,000 years ago, to understand whether it's ideas and material culture that moves in, or do we also with ancient DNA can we detect people or genes moving into this region. So with that, I think I'm really out of time, so I'm gonna stop and.
Thank you so much to our presenters. As I said, we now have a chance to ask a few questions. We have received a couple of really good ones. The first one was for John from Howard Bernstein. How can you tell which sequences are human and which ones are bacterial?
Great, that's a great question. So it turns out human DNA sequences are very similar to one another. If we were to line up the DNA of any two humans across the globe, there would be a difference about once every 1,000 base pairs. And so what that means is we can sequence this ground up bone powder that is a mix of bacteria, soil bacteria and bits of human DNA, get the sequences out and try to line up those sequences to a single reference human genome, like the human genome sequence that was done in 2003. That sequence, we can try to line up our short sequences from the experiment against that one, and any human DNA will line up to it, and any non-human DNA hopefully won't. Sometimes it does, but that's pretty rare, and so that's one of the ways that we tell that we have human DNA versus bacterial DNA. And so we'll find in many of these samples that for every 100 reads that are sequenced, only 5 out of 100 will actually be human, say. And at that hit rate, you're actually pretty happy. You have enough that you can do work with.
Thank you, John. We have a question from Leslie Brown. I'm going to unmute Leslie so she can ask.
Hi, this is actually a question I've always wanted to ask, and this is around racial bias, which is an issue and has been, I would say, for centuries. How is it addressed in archaeology, considering the example of the Kennewick Man, numerous examples, including racial classifications that were specifically created to justify inferiority versus superiority among Europeans and people of color, specifically justifying enslavement? Are archaeologists held to account for their biases within the archaeologist community? And if so, how is this done? Since the implication of population movements is powerful and tells an important and accurate story of history. And then one more thing, those archaeologists who wanted to speak to people of color, for example, Ivan van Sertima, who wrote the book They Came Before Columbus, to show that Africans did come to the Americas before Columbus, but there was a lot of backlash on him to say that. So a lot of questions. Don't know if you can answer them, but love to hear your response.
Thanks, Leslie. Maybe I'll start, and I don't know if the others want to jump in. That's a great question and an area of conversation, really, and thank you for raising it. There are many dimensions to think about. In terms of the Kennewick example, it's sort of where the idea that there are European skeletal features and non-European skeletal features that first created even that initial assignment, right, is tied in a way to I think the history of racial classification and anthropology that you're referring to. And so there are ways in which the molecular work has been powerful in helping advance and move beyond some of those simple classification schemes, and in cases like this, empowering to the Native American communities and helping them reobtain the remains of their ancestors that they have the right to. And but there's a large and complicated history in archaeology in relationship to race, in genetics in relationship to race, and it's a challenging legacy that we have to deal with as a discipline, and that we are trying to do our best to work without that sort of racist framework that some of this science began in. And there are many ways in which that's trying to take place. Within the human genetics department, we actually have a reading group around the topic of genetics and race, and are trying to engage these issues and incorporate them into our teaching and our curriculum in ways that 10 years ago wasn't part of how science was taught. I think increasingly we recognize how the science intersects with society, and our the PhDs that we train, the graduates that we train need to be aware of this history that you're referring to, and the ongoing practices, and make them as sensitive as possible. So I'm trying to say that to kind of give a compressed answer, because I think we could talk about this for a long time. I think it's a very important issue. I hope that helps.
And perhaps I can weigh in just briefly on the same topic. As an archaeologist, the term race, and even what was meant by it by older generations, is actually not used anymore in archaeology and anthropology, because it's so problematic, because it's been so misunderstood and abused. And if we're really talking about various kinds of population level genetic variation, then very often those don't map onto a specific culture or language or way of doing things, or any of the other kind of things that that were assumed when race was kind of essentialized. And I think what's exciting to me about the recent DNA work that Maanasa and others were involved in with the European populations is it really throws up in the air the old European notion that there was a European white race from the get-go, right? That there's a fairly recent mixing of quite disparate population groups in historical time that produce the pattern of genetics even in Northern Europe today. So that, if anything, that should really cause people to rethink the old notions of race. And John and others in genetics, I know, are extremely careful in how this is addressed. And having said that, what's exciting to look at is that there were movements of populations that obviously had a basis of cohesion, that had some similarity one with another. They moved together for various reasons, and then that produced all kinds of effects, both culturally and genetically, in the places where they arrived. And I think we can talk about that without throwing into the mix the negative or the problematic race concepts of the past.
Thank you.
Thank you. Thank you. Thank you, John. Thank you, Maanasa. Thank you, Dr. David. I think we can go ahead. We've been getting really good questions, but we want to give an opportunity to our next two presenters to speak, and then we will go back at the end and ask some of the questions that are coming in. Thank you so much, everyone. Dr. Schloen?
Okay, so I'll go ahead now and share my screen here. Oops. Can everyone see that? I hope so. Yeah. So I want to say a little word, first of all, a word of thanks to Andrew Peters for putting this together. I got to know him last year, as Andrew said. He came on the dig with Sandy and me. We had a great time and found a number of areas of mutual interest. And then I want to thank also Natalia and the Global Engagement Alumni Association team for helping to put this together. I'm very happy to be able to talk to such a wonderful, diverse group of alumni and people affiliated with the University of Chicago. And as an archaeologist, I want to say that what's really prompting this is a unique kind of session that we're part of right now, with a really in-depth engagement between practicing dirt archaeologists who dig stuff up out of the ground like me and the geneticists, because historically there's been very little close collaboration and exchanges at a deep level between these communities. It's a real shame. And one of the reasons for the lack of collaboration and communication is that the exciting and amazing advances just in the last 10 years in DNA research have totally messed up the archaeological consensus that people had, written books about. And this notion that there were, punctate events or population movements that really had a major impact had been dismissed for a while among archaeologists, who became a little bit leery of so-called diffusion models and, leery of migration models and in favor of a kind of slow diffusion model. But now we have to take into account how peoples of the past did move and that had a major impact. Now, when people are moving from place to place, archaeologists are familiar with that phenomenon. They do different things in different places. They develop different styles of artifacts and culture. They leave different kinds of remains. And there's a tremendous diversity both in space and in time, both geographically and chronologically. And now, taking off my archaeology hat and putting on my computational hat, because I started out in computer science and have always been interested in this, I want to say a little bit about the computational platform at the University of Chicago that we've been using and that has great promise for collaboration with human genetics. And really, to hammer home the point, which I'm sure you've already realized listening to John and Maanasa, the University of Chicago is a world leader in computational data analysis and data visualization in many disciplines, and certainly in human genetics and ancient DNA, where just amazing work is being done using the latest statistical and computational methods to analyze, answer questions, visualize data. We have a research computing center, which runs our supercomputer, has a great staff that we work with all the time. It's a central facility, and that's great. But what about data management? And as someone who's been into databases for a long time, there's a real lag in most disciplines, lagging behind analysis and not paying enough attention to the problems of actually managing and querying and manipulating the data itself, especially for the kinds of data we deal with in archaeology and in ancient DNA, namely diverse data from many sources recorded in many ways, sparse data where you have a lot of incomplete cells in your spreadsheet, a lot of varying levels of detail from one data set to the next, and hierarchical data. And by that I mean information that's organized in hierarchies of space, like regions and subregions, or temporal hierarchies, periods, subperiods, relative periods and subperiods, or linguistic hierarchies, getting into the grammatical analysis of text. So those features of the kind of data we work with in many scientific fields and in archaeology means that flat file methods of databases or relational methods, which are very common in the business world, are not terribly well suited to what we want to do. And that's why the OCHRE system, Online Cultural and Historical Research Environment, has been developed over the last number of years. I don't want to get into too many technical details. Some of you may be interested and can ask about it later. But for now, I'll just very quickly mention a few key features. This is a non-relational graph database platform, if you're interested in that concept. What that means is you have a flexible graph of data or network of data, atomized data items not based on rigid tables. It can support many ontologies, many ways of describing the data. It's very highly optimized for hierarchies of data. And Sandy will show you, Sandra Schloen will show you some examples of that in a minute. And we developed it as a platform that would support all stages of the research, acquisition of the data, integration, analysis, publication, and archiving, the 5 main stages of research, with a technical support team headed by Sandra Schloen to provide training and legacy data conversion. So that's what we've been working on for some years, coming out of the archaeological side of things. And I'll just show you a few examples of this system in action, and then I'll talk a little bit about the genetics potential for this computational platform. This is the site of Tel Kaysan, a large mound, ancient mound in Israel. And if you look at the team photo from last year, you can probably see Andrew Peters. He's got a red circle around his face. He and his son, Kevin, joined us last summer, and we had a great time with them, and a bunch of UChicago students joined us as well. So we use OCHRE in the field. Sandy has developed all sorts of wonderful features for making it suitable for on-site data recording. OCHRE, this platform, is used by textual scholars looking at ancient documents. It's used for geographical analysis, mapping. You can see this high-resolution visualization from Iraq that was generated out of the system. And most recently, it's been used in a project with our computer science department to do a machine learning AI type project to automatically read cuneiform tablets, which are very hard to read, even for modern expert scholars, but using the latest techniques of AI, deep learning, neural networks in order to get that done. But returning now to the point of contact between me and John Novembre and Maanasa, this has to do with the potential of this data management platform for facilitating data science in general, but also specifically ancient DNA data analysis of the kind that John in particular does as a computational biologist. So the system, the OCHRE platform, is integrated with an R server, a statistical computational server, a very high capacity, high powered system that can be used for data analysis and visualization, and removing a lot of the awkward steps and the cumbersome steps that would historically have had to be done to prepare the data for analysis, making it a really fast and smooth iteration between selecting the data, running the analysis, viewing the results, and then rerunning a new analysis. Here's an example of a network analysis plot from an analysis of thousands of ancient Greek coin hoards studied by a colleague in our classics department. But I'll finish my part of the discussion here by emphasizing the degree to which ancient DNA specimens and the genomic reconstructions that John and Maanasa and others are doing really fit with our platform. They come from many geographical locations in different periods of time. Again, diverse data from many sources, sparse data, hierarchical data, all these things are true of ancient DNA. And the OCHRE platform that we developed independently of the genetics people turns out to be really well suited for their kind of data. and is something that we want to develop further as a platform for genetics research in a way that hasn't been done before. This is a real cross-disciplinary initiative, a unique collaboration, really not being done anywhere else that I know of, that can be done here in Chicago now that we have such a world-leading, top-notch human genetics, ancient DNA facility. And also, for getting back to the archaeological interests that I have, this could be a great way to really move ahead with cutting-edge research about ancient peoples and migrations. In my case, the Middle East is where I work. The ancient Middle East is my cup of tea. That's what the Oriental Institute in the University of Chicago is all about. But there are so many questions about the development, the origin and development of human civilization in the first place, 5,000 years ago, that have not been answered and could be answered with ancient DNA research, properly analyzed with powerful computational tools. And I'll just throw out some questions. Who were the Sumerians? We don't really know anything about them genetically. These are the people from ancient Mesopotamia who created the first cities and invented writing, the very first written culture in the world in southern Iraq. Who were the Canaanites of the Bible? This is a famous group, the Phoenicians that I'm studying now. I'm trying to start an excavation in Spain, looking at the Phoenicians from the Eastern Mediterranean who had sailed far to the west looking for silver and in Spain. This is an exciting area of research. Well, these are a branch of the Canaanites, but how are they related? And the Israelites of the Bible, how are they related to the Canaanites? A lot of people now think the Israelites are really just a branch of the larger Canaanite group. So those are the kinds of questions that I would hope to answer in collaboration with our human genetics colleagues. And I'll leave you with this slide here for a moment. With a particular exciting possibility that just came up recently of studying skulls, 5,000-year-old human skulls excavated by Chicago archaeologists in southern Iraq at Kish. You can see the red arrow pointing to where Kish was in ancient Mesopotamia, one of the first major cities of the Sumerians. One of the oldest records of ancient writing was found there. These skulls are in Chicago. They're accessible for DNA analysis. We just need to get a project together to do that in order to understand the Sumerians, first of all, the originators of our civilization, the first people to invent writing and cities and so many of the technologies and ways of thinking that we as latter-day urbanites have inherited. So I'm going to stop there with my presentation and turn it over to Sandra Schloen to talk a little bit about the details of using this computational method for ancient DNA work. And then we'll have open it up for all sorts of questions and be happy to stay a while and talk with all of you. Thank you.
So when John Novembre first came to us at the Oriental Institute, his team, tired of wrangling diverse and disparate data sets in order to do their analysis, the prospect of working on an online, collaborative, integrated research environment was appealing to them. The opportunity to work with scientific data, precise, well-formatted, and standardized, rather than the fuzzy and idiosyncratic data sets that we typically deal with in the humanities, was appealing to us. Having been involved in data management for archaeology projects for over 20 years, this was right in our wheelhouse. Whether documenting ancient Greek coin hoards scattered around the Mediterranean, identifying looters' pits disconcertingly abundant in central Iraq, or tracking harpoon blades unexpectedly littering the Central Sahara, we specialize in managing highly descriptive data contextualized in space and time. Indeed, OCHRE was already cataloging human remains, which are common sources of DNA samples in historic context from central Niger, from a Peruvian valley, and from the coast of Israel, along with tens of thousands of faunal remains, itemized, identified, measured, and photographed from excavations all over the world. We agreed to do a pilot project in OCHRE, and John supplied us with 5 spreadsheets which had been published as supporting data for 5 articles in the journal Nature, all 5 spreadsheets in different formats. At first glance, the data was clean and well formatted, as we expected, but as we worked with it in detail, no offense to our scientific colleagues intended, it was a bit surprising to learn that they are as prone to the same problems of data variation and the lack of coordination and compatibility as our colleagues in the humanities. And so we set about doing what we do best, data representation and integration. As David has already mentioned, OCHRE uses an item-based approach rather than flat files or tables to represent data. Plucking each row from the constraints of its table, we create a new OCHRE item, being careful not to duplicate items or create redundant items if they are sourced from more than one table. OCHRE gives the item a name and a universally unique ID to avoid confusion with any other OCHRE item that might have the same name. But what of all the original names of the items from the original tables? You'll notice that the sample IDs were called many different things, and even at a basic level, aligning such tables for analysis would be problematic. From our archaeology background, we are used to items having different names, ancient or historic, English, Hebrew, Arabic, French, or German, and maybe all of the above. OCHRE's alias mechanism and multilingual features let us assign any variety of names to the same item and treats all those names as if they were equivalent. Problem solved. What of the data differences between the 5 different sources of information? 45 of the almost 2,000 samples were treated by all 5 studies, most of them treated by more than one study. Again, our archaeology experience had this covered. Picture 2 archaeologists standing over the same shallow circular pit. One calls it an impressive oil press installation. Another calls it a latrine. Establishing each source of data as an authoritative person item in the database, OCHRE allows potentially multiple observations on a single item and attributes each observation to the original source, being careful to give credit where credit is due. Problem solved. Each observation contains a selected set of what we call properties, descriptive variables assigned corresponding values to describe the details of the item. These can be customized by each project as needed. Only properties relevant to the specific item are used. There was wide variation in how the samples were described. Take, for example, this tooth specimen, being noted as simply a tooth in 3 of the 5 sources, but one added that it was a premolar, another that it was the right upper premolar tube. OCHRE's descriptive properties are hierarchically organized with higher level descriptors encapsulating more detailed ones, with the more detailed descriptions inheriting from the higher level ones. Tagging the tooth as a premolar tool, it inherits the notion that it's a premolar, which in turn knows that it's a tooth. This way, we don't lose the details, but we provide compatibility with data not presented at the same level of detail. While these DNA specimens were also located spatially, there was a wide variation in how geographic places were noted. Matching on these spatial contexts to align spreadsheets was problematic and in fact required some cleanup even to rationalize them in OCHRE. But between the use of aliases to accommodate multiple versions, abbreviations, and spellings of the names, and the use of hierarchy, again with the benefit of inheritance to accommodate different levels of detail, we recovered. Whether the specimen came from a cave, within a site, within a region, within a country, or whether we don't know anything more than this was found in Hungary, every item finds its appropriate place in space. Ditto for temporal context. A detailed hierarchical taxonomy was needed to capture the variations of dating schemes used on the ancient DNA samples, these being designed in collaboration with our genetics colleagues. But other period-based hierarchies also capture cultural and historical context for the specimens. Note that the historical chronology in the top right is shown mostly in gray, indicating that it is borrowed directly from the OCHRE Master Project as it is a common historical framework, Neolithic, Chalcolithic, Bronze Age, Iron Age, shared by many of our archaeology projects. For example, UChicago's Paul Sereno's Goboro project uses the same chronology to tag the accordion man and the other skeletons from his site. This avoids redundancy and reinvention, giving new projects a head start when they can borrow existing structures already available in OCHRE. In an effort at comprehensiveness, OCHRE allows integration of many other kinds of data, including bibliographic references, linked open data with the World Wide Web, and here I'm showing Iceman from Wikipedia, and geographic coordinates, to name a few. A quick trip directly from OCHRE's Coordinates tab to Google Earth reminds us why we call him Iceman. New to OCHRE for this project was the detailed, hardcore scientific data. Here, a sequence of a genomic library, those A's, C's, G's, and T's continuing off the screen in a very long list. And despite having passed organic chemistry in college, I don't understand many of those details, but in collaboration with Maanasa and John, any data they want to generate from their research or from their lab, we'll find a proper place for it in OCHRE. Having captured all kinds of data in a rational and integrated way, OCHRE provides an extensive query mechanism to select relevant subsets of the data using many kinds of criteria, including matching on descriptive or metadata properties or character strings, and scoping by spatial or temporal contexts. Here is a sample query requesting just female specimens derived from teeth. The resulting list of query results can be presented in any number of ways, including, most commonly, tables. At which point our scientist friends breathe a sigh of relief, reassured by the familiar format. So are we back where we started? Yes and no. Yes, because we didn't want to lose anything in this process and could essentially recreate the data from which we started, only cleaned up. And no, this time the data is based on items, contextualized and integrated, ready to create many different kinds of tables and primed for easy reuse for many different kinds of analysis. Much better than what we started with. Here, for example, is that same table of data from the same query result, but viewed instead in OCHRE's built-in interactive map environment with access still to all the other item data in the database. People assume that as a highly flexible and comprehensive database platform, OCHRE must use a complex data model, but it couldn't be simpler. Items of all kinds, organized in hierarchies, which are user-defined, including hierarchies of space, time, and description. Items described by properties, which are user-defined and integrated in a common framework, ready-made for analysis and visualization of all kinds. Our proof of concept confirmed that this simple strategy works well for the management and analysis of even complex scientific data, and we look forward to ongoing and fruitful collaboration with our colleagues in genetics across campus.
Thank you so much, Sandy. Thanks to all of our panelists today. We have a few really good questions. The first one is from Naveen Gupta, and he asked us, the surprising amount of DNA mixing suggested earlier start to civilization than is conventionally accepted today. And this is for any of our panelists to ask.
David, can I let you go on that one?
I'm sorry, I didn't quite catch the question. Could you repeat it one more time?
All righty. Does the surprising amount of DNA mixing suggest an earlier start to civilization than is conventionally accepted today?
No, so if by civilization we mean the development of very dense populations living in cities, even walled cities, and beginning to invent writing, as a bureaucratic mechanism of administration, that's very securely dated based on radiocarbon evidence and based on when these cities first show up. So what the DNA evidence tells us, though, is it tells us something about the people who first built those cities. Where did they come from? Were they people who had moved recently into southern Iraq near the Persian Gulf? Were they people who had lived there a long time? Was the initial population that created these cities the result of a kind of exciting mixture of new people coming together from different origins and then getting ideas from each other, which could well be the case. These are questions we could never have answered before without being able to look at the genetic makeup of the people in ancient Sumer, for example. And that's why we're so excited by that when we found out they have these skulls from Kish, from Iraq. It's very hard to get samples out of Iraq or other countries, and therefore we do have materials ready to go if we can get a project together to analyze them.
Thank you. Karina Melnick, you also have a question. Go ahead and unmute yourself.
I messaged you one question, but I have a different one in mind right now. Is that all right?
That's perfectly fine. Go ahead.
Okay. Well, I'm very interested in the process of sort of deciding that a DNA sort of sample belongs to a certain population. So Dr. Schloen, you were talking about how that there's like some hints that the Israelites were just an offshoot of Canaanites. So I'm wondering if, let's say, you have a limited amount of skulls or of DNA, how do you say, or how do you get to the point of saying that, okay, I've determined that these people were Israelites and this is what the Israelite DNA consisted of?
Right. So first of all, there's a problem of defining what we mean. If by Israelite we mean people who lived in the region that was the kingdom of Israel in biblical times, then you're right, the more samples, the better. And there was a recent study done that included a few dozen specimens. Now we want more than that. But if I understand correctly, and I think this is where John can also correct me as a statistician as to what would be a plausible number of samples to analyze, even a few dozen samples from different locations within the region of interest can tell you a huge amount about the genetic makeup of the population at that time. But maybe I'll leave it to John to elaborate a bit more.
Sure. So I'm going to answer the question in part by telling a little bit about this project that we conducted in Sardinia, where one of the hypotheses about Sardinian population history is that it's been a place of substantial isolation in terms of the population history. There's evidence of cultural contact, of archaeology, of pottery and so on, that from the broader Mediterranean, but genetically, Sardinians have a kind of unique genetic signature. You saw on that map of Europe, they're sort of off, sort of these 5 samples off to the side. And so in looking at a time transect of Sardinian history, we were able to get around 70 samples from different periods of Sardinian history. And we were able to see how there was a continuous line of ancestry in Sardinia from the early founding, that then, but then there's a period where they receive new ancestries. And it's the intersection of the archaeology and the genetics that allows us to understand that, in the Iron Age, we start to see these individuals on the island of Sardinia who look much more like Eastern Mediterranean individuals. And those individuals are buried in Phoenician contexts and Punic contexts. And so it tells that intersection is what allows us to see, okay, Sardinia at that time becomes a place with immigrant ancestries, and then we start seeing ancestries that look like they're coming from Spain and from Italy. And we see that Sardinia becomes a more like cosmopolitan place in a way. And so it's that kind of looking at the genetics, how they spread on those PCA maps, and then intersecting that with the archaeology, where you start to say, oh, so things have changed here. And there's new ancestries that are shifting the composition. In terms of sample size, it's interesting. There's sort of one view is, well, if we sample just a single individual, like my DNA comes from my 2 parents, my 4 grandparents, my 8 great-grandparents, my 16 great-great-grandparents. So in a way, I carry the DNA, if we go like, 8 generations back, some like 2 to the, 32, 64, individuals, DNA are carried in me. So just my bones would tell about a community in a way, right? But at the same time, in diverse societies with heterogeneous ancestries, you know, just having one wouldn't be enough, right? And so, so while some of the first ancient DNA studies got a lot of information out about it just from 2 or 3 individuals, we already learn a lot, because that even like I said, even just one individual can tell you the history of many. Like in Sardinia, if we had only, there are certain sites where if we only look in like on the island at the same time, there are Phoenician colonies and inland populations. And if we only sampled, you know, one individual, you know, and not from both places, we would miss out on complete stories. So larger sample sizes do help, even though we can make a lot of progress with just a few.
Yeah. And this is such a great question, and I wanted to say something more because I have the map up now for all of you to see of ancient Mesopotamia. So if you're thinking of these early civilizations that have been so influential, the Sumerians in southern Iraq, other Mesopotamian cultures, the Babylonians, the Assyrians, well, this gets us into biblical archaeology, which is something I'm interested in. And the fact is that the Bible would lead one to believe that the Israelites are descended from Mesopotamians, right? Abraham, Isaac, and Jacob, they come from Mesopotamia. But you know, most scholars, most modern scholars, think that that's somewhat legendary, and that the vast majority of the people who claimed an identity as Israelite in the Bible, their grandparents were Canaanites, were pagan Canaanites. So it's really an interesting archaeological conclusion that has never been able to be, we have never been able to test it actually from DNA. And that's why it would be exciting to combine the recent DNA studies in Israel, where a lot of DNA work is being done, Lebanon, Western Syria, compare those populations to Mesopotamian populations, and then we'd have a much better sense of these early populations in the Middle East who invented farming, right? When we talk about the Neolithic farmers who moved into Europe and added their addition to European DNA, that's where they're coming from. And so I think there's a lot to be done. It just tells you how much we don't know at this point.
Thank you so much, Dr. Schloen. We have another question. Bjorn Telman, I'm probably mispronouncing your name. If you would like to go ahead and ask your question.
Sure. Can you hear me?
Yes.
Yeah. So first of all, thank you for what I think has been a very interesting evening. And I guess my question is just really, where do you see the field heading in the next decade, you know, given the advances that you've highlighted, and what do you think are the most promising possible developments, and what questions do you think they might answer above and beyond the ones that have already been raised?
I'm happy to take that. At least I can start off, and then the rest of the panelists can feel free to jump in. So interesting question, and luckily we're not yet at the saturation point of the plot that I showed during my presentation. I think there's a lot of unanswered questions, both when we talk about human population histories, which has been the focus of this presentation. I think one of the big things is thanks to the technological improvements that we're making in the field, just generally within the field of biology and molecular biology, but particularly in terms of leveraging some of these difficult samples, particularly from what were once considered tough environments, which are primarily the tropical environments, which is also why the map that I showed you is very much skewed towards, the data skewed towards certain regions, temperate, the Arctic. And so there's a lot of excitement now to move back closer to the tropics and kind of try to fill in the gap. And of course, one of the big regions is, as Professor Schloen said, the Near East, the Fertile Crescent. A lot of the initial migrations, including the advent of agriculture and spread, has happened out of there. And so the ability to now get in there, revisit, in fact, some of the samples that maybe 10 years ago were not possible in terms of ancient DNA because the technique and the technology wasn't there yet. So to sort of start filling in a lot of the gaps that we have in terms of the temporal and the geographical range when it comes to humans. But I also wanted to quickly add that in addition to making progress in terms of getting access to DNA from samples that maybe didn't work a few years ago, we're also making progress in being creative about the sources. So a couple of things that I don't have time, but really excite me, particularly as a researcher in this field, is the ability to use things like sediment DNA. And so there have been a couple of studies that show that, whether it's humans or animals or even plants, we leave traces of ourselves, our DNA over time, and this can get preserved, again, depending on the environment. And so in the absence of fossils, you can potentially use proxies, inorganic proxies like sediments or ice cores, or even I saw a question about, someone asked about marine, and so marine archaeology and sort of coupling ancient DNA with that also opens up, again, the sort of treasure trove of sources that one can actually use to study the past. So those are some of the things I think that excite me, but I'm going to also let the other panelists chime in.
I'll highlight one area that interests me, which is being able to look at the histories of particular genes and how they have spread. And so in particular in Sardinia, there are high rates of beta thalassemia. And this is a type of anemia that is a disease, but it is the same gene that gives rise to this disease condition also confers malaria resistance. And so how and when did it spread in Sardinia? It's a kind of medical archaeology, a kind of biomedical, like understanding the particular constellation geographically of these conditions. Can we begin to understand them through the lens of human history and evolutionary history? So with these tools at the moment, we tend to look at across the whole genome at many locations and build maps of genetic relationships amongst individuals. But what if we focus on a few locations and tell the stories of particular mutations? Where did this mutation arise? How did it spread? How did it get to this location? And I think that will be interesting in terms of understanding the history of particular biomedical conditions. So that's one area I'm interested in.
And can I just add to that? I'm glad you brought that up, because also in terms of, again, combining the health aspects with archaeology, one other really interesting avenue, I think, is the ability for us to study ancient pathogens. And so a lot of these human bones, especially if the individuals died of an infection, particularly bloodborne infections, we now have the ability to retrieve and analyze at the same time both DNA from the human themselves from the bone, but also potentially any infectious agents that they carried. And so in addition to again, as Professor Novembre said, we could sort of trace the histories of diseases, but both non-communicable as well as communicable. And I think that's also providing some very exciting insights into how epidemics panned out in the past and how some of these infectious diseases have evolved in terms of their virulence over time.
Thank you, Maanasa, John, David, and Sandy. And I want to thank everyone, because we are kind of like at the end of the timing that we had scheduled for this. However, I know some of our panelists have agreed to stay on and take more questions. We will gladly have you on for as long as you want, or as long as you can. I want to hand it over to Dr. Schloen for a few last words for those that need to jump off the call.
Thank you, Natalia. Once again, this has been done so professionally, and the staff worked to set this up, and it was wonderful to see so many people attending and asking questions. Clearly one reason that we as researchers are presenting this to you is to let you know, here's a real opportunity for groundbreaking research that, to be honest, is very hard to find funding for from ordinary granting sources. So here we're talking about work at the intersection of two very different worlds, the world of molecular biology and DNA and genetics work, and the world of archaeology and ancient history and ancient culture. And there's been a lot of amazing progress. It hits the news all the time. I don't know how many New York Times articles I've read in the last few years that are reporting these things, but it's actually woefully underfunded from the point of view of federal granting agencies. And so I think it's just worth everybody knowing that here's a way that private philanthropy can really make a huge difference, that it's not something that, that is being as well supported by public funds as one would hope. The other thing I want to say in closing is that, and I realize this may sound insincere, and I really want you to believe that I'm sincere when I say it, is I cannot think of a better place than the University of Chicago for this kind of work. The culture of crossing the aisle, going to another department, meeting people from completely different parts of the campus, over and over again. This has happened to me over many years, and I've had wonderful collaborations and projects that have developed out of that, and it's been easy because the culture of the place is really special, that it encourages interdisciplinary work like this. And the fact that John and I and Maanasa could so easily get connected, even though they're across the campus from me. So I just wanted to put in a little plug or a little praise of our university that we're all affiliated with. Many of you have earned degrees there, because it's really a special place. And I cannot think of another place in this country where the collaboration between geneticists and archaeologists is developing in such a robust and sophisticated manner as is happening here. So with that, I can just thank you all once again, and I'm happy to stay. I know John and Maanasa probably are as well, to stay for a while if there are more questions, just to chat.
We do have several questions, and I'm going to try to give the floor to David. Maanasa had a couple of questions for the panelists. David, if you want to go ahead, unmute yourself if you're still with us. You can unmute yourself and ask your questions, or I can do so for you.
Yes, thank you very much. Can you hear me all right?
Yes, we can. Yes, we can.
Thank you. So the one question which Sandra has also has already answered, but I was kind of asking how, in terms of the computer program, how they differentiate between the quality of the inputs coming in from the various sources, and you know, are they treated equally? And that was one question. And then another one that came up as I was thinking about this. You know, the studies that were so famous for a long time of Dr. Louis Leakey in Africa, it seems to me would lend themselves to this kind of study. Is there any thought to do anything with those studies? Thank you.
So maybe Sandy, you could answer the question about the computer question.
I'll just comment on the first point there, that the point of trying to be careful about attributing the source of the data is that we don't get this notion that the computer says something authoritative. We want to make sure it's clear to the scholar where the data came from. If they don't agree with somebody, if they don't respect that study, if they know that there were circumstances under which it was done poorly, a query would let them filter out data that they did not want to include in an analysis. So OCHRE as a system tries to be completely neutral and agnostic, but give the scholars as much information as possible so they can make valid decisions about their own research.
For the second part of that question.
Africa. John?
Actually, Maanasa, would you want to handle that one about sort of the use of ancient DNA for deeper time samples? I think most of the examples we've focused on have been from the more recent past of the Paleolithic and onwards. But yeah, go ahead, Maanasa.
Yeah, so I think this again relates to some of the comments from the previous question addressed. So Africa, again, much like some of these continental scale regions that have no or very little representation, if you asked DNA researchers maybe about a decade ago whether you pick up samples from Africa and work on them, most would look at you and laugh, because at the time it was pretty much a given that the tropics just simply wouldn't work. But really what's turned things around the last 10 years is the ability for us to both recognize the limitations, but also leverage some of the recent technologies that molecular biology as a field has laid out for us to adopt in ancient DNA. So consequently, we've had some amazing genome-wide data sets from Africa that have come up in the last about 3 or 4 years. I believe some of the oldest material or DNA data from Africa is about in the last 8,000–9,000 years. So it's deep, but it's not deep enough. And someone mentioned Louis Leakey and some of course, the other famous paleoanthropologists, but temporally, there's also a limit because DNA does keep degrading over time. And so the sort of theoretical limit to how far you can go in terms of deep time is about 1 million years. But the 1 million years is also the sort of the most pristine environment that you can get, which would be the Arctic or the Antarctic, which are basically freezers that will preserve DNA for a long time. But otherwise, it really comes down to the environment and the preservation. So it's likely that some of these old fossils from Africa may not lend themselves to genetic analyses much. But that being said, some of the fossil evidence from, let's say, temperate Europe, for instance, we have Neanderthals, we have even further back, some of the oldest materials about 450,000 years from some of the sort of other hominid sort of species or subspecies that have been sequenced. But particularly coming back to Africa, unfortunately, the environment doesn't lend itself to going back to some of these very, very old fossil finds. So that would be the limitation. I don't know if that answers that particular question. If there was anything else in terms of details, I didn't really catch the entire question.
Hopefully. Thank you, Maanasa. We had a question from Seema. She submitted a while ago, and another from Susan. I think Seema's question has kind of been answered a little bit. Susan was asking, Susan Ware, do your resulting migration maps mirror or challenge linguistics maps of how language families spread?
Mm-hmm. Great. So yeah, I'll answer that one with that map of Europe result that I showed. One reaction to it was, well, isn't that kind of just what you'd expect, that it's geography? But yet, if you were a linguist, you'd say, oh, this should have been in groups of the Romance language speakers, Germanic language speakers, Slavic language speakers, and sort of more clustered by language. But it does appear very geographical. And so in that way, at the broad scale, it doesn't, cluster in the way that a linguist might expect. But there are individual stories that one can look at that give it finer resolution. One of them is the Hungarian samples actually also look like their geographic neighbors, whereas Hungary is a linguistic isolate in the sense that it's a Finno-Ugric language, not an Indo-European language. So in that way, and for that map of Europe, it does sort of not really align with how linguists would have thought it would play out. And then there's a broader story that's been interesting about the deeper time origins of Indo-European languages. So one of the major ideas was that Indo-European languages spread with farming in the Neolithic period, and that earlier samples from the Paleolithic would have been the pre-Indo-European speakers. And a number of different lines of evidence from ancient DNA and the way that it overlaps when intersected with archaeology and studies of samples from, for instance, the Basque and Sardinia as interesting European population isolates point to how it's more likely that the early farmers were speaking a pre-Indo-European language, and that it spread with these steppe populations into Europe. And so that's quite fascinating, quite surprising, because that was sort of more of a, David, you can come in here, but was more of a fringe hypothesis amongst, within a debate, and it now I think has gained a lot of support and precedence. It's still, ultimately we're studying ancient DNA. It's not, we don't get to see what the language the bones were speaking, but these become new lines of evidence in an integrative exercise to reconstructing the past.
And actually, just to look at the complex interplay between movements of groups, you know, en masse, like the people from the steppes of southern Russia with their horses, you know, sweeping across at a certain point, to compare those kinds of movements with language differentiation, it's fascinating because at one level, we all understand anyone can learn any language. If that's the prestige language or the influence, it can be your mother tongue pretty quickly, and you can forget whatever language your ancestors spoke for thousands of years. So language can jump across genetic barriers like crazy. But of course, in ancient times when you don't have jet planes and trains and people moving or TV, you know, it's true that within a given region, if there's not a lot of migration after that first event, then the population might be genetically closely related and then developed a certain branch of the parent language that's specific to that geographical zone. So it's kind of common sense if you can see how these things could go together, and then you could get both results. But as John said, the so-called Anatolian hypothesis that the Indo-Europeans came from Turkey, what is today Turkey, that's blown out of the water now by the more recent DNA work. And actually we're back to a much older archaeological view of the steppes of southern Russia as the Indo-European homeland, which was out of fashion for a while, but really apparently not, it didn't go out of fashion for a good reason. So that's why the archaeologists get a little hot under the collar saying, oh, these geneticists are now correcting us, but we should be happy to be corrected and to integrate the data with our own evidence.
Thank you, David. We have a question from Ayala Work Awoke. If you'd like to unmute yourself and ask your question. Let me see, or I can go ahead and do so myself. Let me see, let me try. Okay, I will ask, any future plans to study and analyze the African continent since it is the origin of mankind? Take Ethiopia, as Lucy is believed to be the oldest human. Also, in the Bible, as Ethiopia has been mentioned over 50 times.
Actually, the first genome out of Africa came from Ethiopia, but we're definitely not pushing the temporal limits that the question addresses. Again, going back to the preservation conditions, you know, Lucy, we're talking about millions of years, and that really predates the sort of even the theoretical limit to DNA preservation. So that's sadly a dream that's going to remain a dream for geneticists. But on the other hand, you know, even unlocking the last 10,000, 20,000 years worth of prehistory of Africa is still a lot more than what we thought we could do, practically speaking, 10 years ago. And so there's a lot of work now to both, you know, in sort of developing or generating data sets from Africa, particularly, again, addressing questions around movements of people relating to, let's say, the spread of farming, related to the Bantu spread, and even understanding, again, the dynamics between farming and hunters-gatherers in the continent, relating to spread of languages there, very much paralleling some of the studies in Eurasia. So there's a lot to be learned even about sort of the more recent human evolutionary history in the last few tens of thousands of years. But with the fossil work, again, it's probably not feasible given the limitations of preservation.
Thank
For
you,
perspective,
Maanasa.
Maanasa, what's the record is held by a horse?
Yes.
Horse
Yeah, so the record for the oldest DNA ever sequenced is actually from settlements. And this is about, it actually hits the 1 million mark, 1 million years. But if you talk about DNA from a piece of bone or a skeleton, then that's an equator, sort of ancient form of horse. But both these record holders come from Greenland and Siberia, or actually not Siberia, sorry, from Yukon in Canada. And so again, it sort of hits home with the fact that it has to be very pristine and like a freezer environment to even preserve DNA that's that old. And then when we come back to the temperate and the tropic, that age range just shrinks to the last few tens of thousands of years.
Thank you, Maanasa. We do have a follow-up question from Seema, specifically for John. She wants to know about the impact of epigenetics in mapping historical populations.
That's interesting. So in general, epigenetics is not something that we look at that much. It's hard to read the DNA to begin with when it's degraded in the ways that Maanasa described. That said, one type of epigenetic mark, which is a methylation mark, that can you can have the basic DNA sequence of A, T, C's, and G's, but some of those A's and T's and C's will have a methyl group on it. And there are some groups that have been able to read methyl marks on ancient DNA. And it's not that it's so useful for learning about population history and how individuals relate to one another, but methylation marks indicate where DNA is being silenced. It's not being expressed in terms of, if you think of our DNA as like a recipe book and our body is reading different recipes at different times, the methyl marks say that like we're not reading that part of the book right now, like the RNA is not being transcribed. So it tells us something about gene regulation, gene expression. And Maanasa, correct me on this, but there have been groups that have tried to use the methylation sequences that they've observed in ancient DNA samples to make comments about gene regulation. And I think possibly even for Neanderthals at this point, but I know there was some sort of mid-range human samples that a group was trying this on. It's very, very like cutting edge to the point of almost being fringe, that like it's hard to believe that you can actually do it right because you're just trying to squeeze information out of very limited data. But yeah, Maanasa, I don't know if there's anything to elaborate there, but yeah.
Yeah, I mean, I'd echo what you said. I think at this point, it's just exciting to be able to even get access to that sort of data. So I think we're still in the sort of, almost patting ourselves on the back that we've been able to do this. So we haven't really progressed to the point where we have enough data even to be able to compare to other individuals and populations in the past. So getting data from a single individual is not going to tell us much. So things like relating to the health or all sorts of biological and functional sort of evolution or even environmental impact. And I think we're still a little far from those sort of overreaching conclusions. But at least we know, technically speaking, it's possible to reconstruct ancient methylomes, as we call them now.
The more exciting, I won't say more exciting, but the more active area of epi research is that we think about the epi community that is our microbiome. And Maanasa referred to this in the sense of doing research on ancient pathogens. One can even get the ancient oral microbiome, because the bacteria living in our mouths actually some of it gets embedded in what's called the dental calculus, especially picture in an area before dental hygiene, the tartar, et cetera, is like encasing bacterial DNA. And so we can sequence this DNA and learn about what bacteria was thriving on the mouths of ancient peoples. And that can indirectly give clues about ancient diets, which is a fascinating line of research.
Thank you, John. We have a question up from Uges. Is there anything new to be said at this point about Northern European paleogenetics?
Great. I saw this one. I think this is a fun question. There have been a lot of studies now of Northern Europe in ancient DNA. So in some ways, I think that the broad brushstrokes, I think, of the story are told, at least at the level of these layers of the hunter-gatherer to Neolithic transition, steppe ancestry coming in, but those are broad brushstrokes. And what is getting very interesting is looking at very fine scales. And looking at like dissecting even cemeteries, burial sites, and seeing who was related to who, and even reconstructing how people related from one gravesite down valley, up valley, and kind of seeing kin relationships at a scale that is really much smaller and nuanced. And I think, and maybe David can speak to this and Sandra can speak to this, like the excitement that archaeologists have, because that's the scale at which they're used to working at. Geneticists are coming in and sampling, you know, a few samples from, you know, broad regions and reconstructing these broad stories, but archaeology as a field has moved to these more micro scale structure studies, and ancient DNA is, I think, reaching them now, or we're kind of moving down to that scale.
I could say if there was a methodology developed for understanding kinship relations in at the community level, this would be amazing, because Northwestern Europe is famous for having nuclear families, and a very big contrast from Mediterranean Europe, Southern Europe, where you have the extended family, where you have 3 or 4 generations in one household, and where, you know, the sons bring in the wives into the paternal household. That is not the way it's been for many centuries in Northwestern Europe, and a lot of people have said, well, this may explain certain things about industrialization or economic development in Northwestern Europe. But the question would be, well, when did that change in Northwestern Europe? I mean, how far back could we see these kinds of kinship relations at the household or village level? This could really change the picture of what you might call the social history of pre-modern Europe.
Yeah, and in principle, you know, the ability of genetics to define second cousin, third cousin, fourth cousin relationships is completely there with clean DNA and the right algorithms. It's the basis of, you know, companies like 23andMe and Ancestry, right? And the getting high enough quality data, having algorithms that are robust to the damage is sort of the challenge for applying and the sample size is large enough are the challenges to doing that in archaeological samples. One piece of progress for my group has been that we've developed an approach that can use really relatively poor quality ancient DNA data to learn about what are called runs of homozygosity, long runs where an individual's maternal DNA matches their paternal DNA. And this happens in small communities or when an individual's the offspring of distant cousins, or well, not so distant cousins, right? And so that gives us an indirect indication of how much cousin-cousin marriage is happening, how small the groups are that mates are being found within, and is in the direction, I think, of the type of more like social history, social processes that David's discussing. So as those develop, I think there's sort of other stories beyond the broad, you know, big group population movements to be told. So great question.
Thank you, John. We have a question from Rob Furman. His question is, and it might have been answered a little bit before, but the Chinese Academy of Sciences has recently published results from analysis of dinosaur DNA. Does this open the possibility of pushing back far earlier human ancestor DNA?
I am not aware of the study. Maybe there's a report, but I would say take it with a pinch of salt. Especially if it's not out after peer review. We're talking solid, 65 million years ago here. So there's quite high chances that, it's probably, especially if it's not published. Again, I haven't read the study if it's already out there, so I can't really comment on.
But haven't you seen Jurassic Park, Maanasa? Wasn't it from?
I was mute myself. Amber.
Amber. I should laugh because maybe they've done it.
Maybe could you speak to amino acid sequencing, because that puts the time date back.
Yeah, so there were definitely reports about amino acids being retrieved from actually 2 separate groups, though I don't remember one of them being the Chinese Academy of Sciences, but I could be mistaken. And proteins do survive longer, and we know that it's more robust in terms of the structure. But even that gets chopped up, so we're not talking about entire proteins or like structural, clusters, but we're talking essentially just basically down to the amino acid level of a few peptide strung together. And there's been reports, but again, they haven't been peer review published. These have come from reports of people, presenting at conferences, and so they've been written up on blogs or just online. So the publications are still forthcoming. So I can't really speak to the quality of the work or the quality of the data until it comes out and we've all had the chance to read it. But proteins, I could keep my mind open to it, but with claims of DNA that's 65 million years ago. Yeah, no comment.
Thank you, Maanasa. And we have one last question here, I think, from Fred Martinson. He asks, how about the makeup of Homo sapiens sapiens vis-a-vis, say, Neanderthals? Anybody? Do you want me to repeat the question?
I was going to see if Maanasa was going to jump in on that one, but yeah, that's been really fascinating. When I went to graduate school, the recent African origin model was with no contact with any archaic populations was what was taught, and there was at that time around that time was the sequence of the first mitochondrial DNA from a Neanderthal, and that mitochondrial DNA lineage. So this mitochondrial DNA is DNA that's carried in, that you inherit only from your mother because it's carried in an organelle. It's not your nuclear DNA. It's a kind of DNA that rides along in your cells and in the cytoplasm, and it's inherited from the egg, and the sperm doesn't contribute any. So it's inherited maternally. There's a much higher copy number per cell, so it preserves well. And this mitochondrial lineage of the single Neanderthal sequence fell outside of all of the mitochondrial DNA of humans. So the tree would look like, let me get from the camera here, is like all human mitochondrial DNA, then a long branch, and then a Neanderthal sequence. And I remember reading a paper that did some probabilistic calculations of what are the odds of seeing this if there was mixture. And so, well, there's some probability that there was mixture, but your best guess is that there wasn't. Okay, and it wasn't until I think around 2006 was the first whole genome sequencing of a Neanderthal individual, and that took place thanks to this revolution in sequencing technology. And with that data, it became clear that the mitochondrial DNA was a sort of not telling the whole story, and that there has been some contact and mixture between Neanderthals and humans. And since then, more studies have come out that have shown that humans mixed with another ancient lineage called the Denisovans, and in Africa there were mixture with other lineages. So there were modern humans are a largely cohesive, evolutionary bunch, but we have had contact with these more distant cousins as we have spread out across the globe. These contacts have not had a major impact in the sense that any one person only has a few percent of their DNA coming from these archaic sources, but the contact did happen, and it's been a fascinating piece of history that we had no idea about to really be able to understand without ancient DNA. Yeah, just I can tell you that there were some hints, but it was really hard to believe any of it until the data came in.
Thank you, John. And we have one last question. I think that it's really good considering, all these modern 23andMe tests. Leslie, go ahead and mute yourself if you want to ask the question. I want to know personally what that means as well.
Sure. So as a 23andMeer, there is a percentage of DNA that they list as unknown. And is this because they've not collected DNA in a specific region at this point? Or is that unknown perhaps an extinct population? So I just would love to know, because they do have a large DNA database.
Mm-hmm. Yeah, I imagine that they're having to, in the algorithms, they're having difficulty assigning it to one region. So it's more about uncertainty. Yeah, what 23andMe is doing when they develop these percentages is really worth explaining. If we think about these percentages, they have to, like, the question itself is not really well specified to say, like, what percentage of my DNA is from Northern Europe, Southern Europe, South America, Africa, etc., without specifying a time frame, right? Because for my own personal history, I have a parent who was in South America and a parent who was in New York. I have grandparents who were in South America and in New York, but then you get to great grandparents who are in Italy and Ireland and, and Germany, et cetera. They all spread out, right? And so at what time frame are we asking this question, right? And we go back far enough, all of us are 100% African. So the question is, you should immediately recognize it's not well specified. They're answering a question that isn't even like properly asked. So the way that I tell people to understand it is to think about what they're doing is closer to what we do when we specify colors in a coordinate system. So when we want to refer to the color blue in the RGB system, that's a bad example, blue in the RGB, so yellow in the RGB system, that's red, green, blue, right? We can describe that color as a mixture of red, green, and blue. And that, with a set of weights on those three proportions of red, green, and blue, that will describe yellow, right? And so it's not that the color literally was made up from a mixture in those portions, but it's a useful description of the DNA that's there. So think about those percentages with a huge grain of salt. They are meant to reflect somehow like where your ancestors were, like roughly 500 years ago or something, but it's not well specified. And so I think it's better to see it as, if they had to describe you as a mixture of things in the data set, this is the portions they would use. And that's why the numbers jump from one company to the other, because they're using different references. It's like as if one company selling paint is using red #1 and another company is using red #2, then describing yellow, you'd use like different proportions of red 1, red 2, right? And so that helps you maybe understand why the numbers don't necessarily agree across companies, why they might disagree, change if you shifted the time scale at which you're looking at. So I hope that helps. But I think really with when they say it's unknown, that's acknowledging that, like, even in the statistical exercise of trying to say like what mixture you're made of, they're saying there's a part that they're not exactly sure how to describe it as of being more similar to one versus another. Yeah.
Thank you, John. I think I've run out of my good lights. This just shows how interesting this session has been, and everyone's still here. I want to thank everybody that joined. We had over 100 people join us today, and it's incredible that even past the time that we had, there's still 37 of us listening to the answers to all these very interesting questions. One last huge thanks to David, to John, to Maanasa, and to Sandy, and to Andrew for making this happen today. We are here at the University of Chicago Alumni. We live for this. This is super exciting. And let us know what you thought about it. I'll send everyone out a follow-up email with the link as well to the recording if you want to go back and listen to anything in specific. Feel free to reply back with any questions that were unanswered, and we'll make sure that we get them to the panelists. And once again, everyone is welcome to any and all of the alumni events that are happening right now virtually all over the world. Thanks, everyone. Have a good night. Have a good rest of your week. Enjoy the rest of the summer. And hopefully we'll see you once again in one of these other virtual sessions. Bye, everybody. Thank you.
Everyone, thanks. Thanks, Andrew.
Bye-bye.